My name is Eliza White, and it is March 9th, 2026. Cells are the basic unit of life, and living organisms are either made of prokaryotic or eukaryotic cells. Prokaryotic cells, such as bacteria, lack a nucleus and membrane-bound organelles. Eukaryotic cells, on the other hand, such as human cells, contain a nucleus and specialized organelles. Even though these two types of cells are very different structurally, they often interact in ways that benefit both organisms. One important example of this occurs in the human gut microbiome, where trillions of bacterial cells interact with the epithelial cells that line the intestine. These interactions play an essential role in digestion, immune system regulation, and overall health. The first cell involved in this interaction is a bacterial cell, which is a type of prokaryotic cell. Bacterial cells are typically between 1 and 10 micrometers, and they are simpler in structure than eukaryotic cells. They contain a plasma membrane, a cell wall, cytoplasm, ribosomes, and DNA located in a region called the nucleoid. Many bacteria also have structures like flagella or pili that help them move or attach to surfaces. Bacterial ribosomes are free within the cytoplasm, and they synthesize proteins that are necessary for metabolism as well as survival. The second cell is a human epithelial intestine cell, which is a eukaryote. These cells line the inside of the small and large intestines. They are approximately 20 to 30 micrometers tall and columnar shaped with microvilli that increase surface area for absorption. They contain a nucleus that stores DNA and membrane-bound organelles, including the mitochondria that produces energy, as well as the endoplasmic reticulum and the Golgi apparatus for protein processing. Eukaryotic cells also contain ribosomes for protein processing. In the gut microbiome, bacteria live on the surface of intestinal epithelial cells and within the mucus layer that protects the intestine. These bacteria help break down complex carbohydrates and fibers from food that human digestive enzymes cannot fully digest. Long fiber chains, such as cellulose, which is a component of plant cells that helps them maintain their shape, cannot be absorbed directly by the intestinal epithelium and must be broken down in other ways. During this process, bacteria ferment these compounds and produce short-chain fatty acids, such as acetate, propionate, and butyrate. These molecules are then absorbed by the intestinal epithelial cells and go into the bloodstream. This is how they are used as an important energy source. Butyrate in particular helps maintain the health of intestinal cells and supports the integrity of the intestinal lining. These interactions also affect the immune system. Intestinal epithelial cells detect bacterial molecules through specialized receptors in the membranes. This communication helps the immune system recognize beneficial bacteria while protecting the body from harmful pathogens that could not be recognized. Because of this interaction, beneficial bacteria help prevent harmful microbes from colonizing the intestine by competing for nutrients and space. In return, the human body provides bacteria with a stable environment and a constant supply of nutrients from digested food. The interaction between gut bacteria and human intestinal epithelial cells is an example of mutualism, where both organisms benefit. Bacterial gain nutrients and a protective habitat, while human cells receive help digesting food, producing nutrients, and regulating the immune system. This relationship demonstrates how prokaryotic and eukaryotic cells can work together to maintain health and biological balance. Studying these interactions helps scientists better understand digestion, immunity, and diseases related to the microbiome.